I'm Rajesh Uppal, Director IDST. We bring sharp insights and foresight on defense, security, and emerging tech. Let's dive in.
Imagine a world where you can print life itself. A world where scientists don't just edit genes, but design them from scratch. Welcome to the frontier of biotechnology, where DNA printing and artificial gene synthesis are reshaping medicine, agriculture, and industry, opening doors to possibilities we once thought were science fiction. Today, over $13 billion fuels the synthetic biology market, projected to almost double by 2030. Why? Because groundbreaking technologies like enzymatic DNA synthesis, compact benchtop DNA printers, and revolutionary genome assembly techniques are making genetic engineering more accessible, faster, and affordable than ever before. Let's break it down. DNA printing works much like a regular inkjet printer, but instead of printing pictures, it prints DNA. Instead of ink, it uses 4 basic building blocks of life, called nucleotides, A, T, C, and G, which are arranged in specific sequences to form genes. Think of it like typing out a custom code that instructs living cells how to behave. Artificial gene synthesis is a process where scientists don't rely on existing DNA samples. Instead, they build completely new DNA sequences from scratch by connecting these nucleotides in precise order. It's like constructing sentences by arranging letters, except these sentences determine the makeup of life itself. Back in the 1880s, DNA printing used chemical methods that required toxic solvents and were slow, expensive, and harmful to the environment. But today, enzymatic DNA synthesis, where biological enzymes act like microscopic construction workers, assemble DNA strands in water-based environments. This makes the process faster, safer, and able to produce longer and more accurate DNA sequences with fewer errors. Thanks to these advances, even small laboratories now have access to compact benchtop DNA printers that can create custom DNA sequences within hours. That's a game changer, turning what once took weeks and massive budgets into an everyday task. The real-world applications are staggering. In medicine, DNA printing helps researchers create custom DNA sequences to study genetic diseases, develop new vaccines, or build personalized cancer treatments that target a patient's unique tumor. During the COVID-19 pandemic, artificial gene synthesis enabled rapid production of mRNA vaccines, showcasing its power to respond to global health emergencies. In agriculture, synthetic biology is solving critical challenges. Scientists are engineering drought-resistant crops that need 40% less water and cereals that fix their own nitrogen, reducing the need for fertilizers. They're also fighting destructive diseases like the TR4 fungus, threatening global banana supplies by creating disease-resistant varieties. And in industrial applications are booming too. Imagine producing spider silk for biodegradable clothing, microbes engineered to manufacture vitamins, or algae designed to capture carbon dioxide for sustainable biofuel production. And as a futuristic twist, scientists are even exploring DNA as a super dense data storage medium, capable of storing up to 215 petabytes in just 1 gram. One of the most exciting breakthroughs comes from the creating method, short for cloning, reprogramming, and assembling tiled natural genomic DNA. This innovative approach dramatically reduces costs and time needed to assemble entire chromosomes, letting researchers engineer powerful yeast strains for biofuel production and disease research. With tools like DNA Script's syntax system and Kilobaser's compact printers, the technology is no longer reserved for giant corporations. It's accessible to small research teams in academic labs, empowering them to innovate faster and cheaper. Scientists are even expanding the genetic alphabet. Beyond the natural A, T, C, and G, they're creating synthetic base pairs that work in harmony with natural DNA but offer more stability, making DNA better suited for extreme environments or industrial applications. Of course, these incredible capabilities raise serious ethical questions. What if someone designs a harmful virus or creates unauthorized human modifications? That's why strict global safeguards, such as automated screening of DNA orders against pathogen databases and engineered biological kill switches, are in place to prevent misuse. Looking ahead, the next decade promises even more revolutionary advances. Imagine printing entire human chromosomes or reprogramming DNA inside living cells in real time. Picture living computers that process and store data, or self-growing pharmaceuticals tailored to your personal health needs. DNA printing and gene synthesis aren't just tools. They're the foundation of a new era in life sciences. But the future isn't only about what we can create. It's about choosing wisely what we should create. Through responsible innovation, global cooperation, and ethical regulation, we can unlock the immense potential of these technologies while safeguarding humanity's future. Because at the end of the day, designing the code of life is not just a scientific marvel, it's a profound responsibility. Thanks for watching. Subscribe, follow, or visit idsth.com for more cutting-edge defense and tech insights. See you in the next one.